Hello. Mine is on.
Is mine on? Ah, there we go. Now it's on. Hello, everyone.
Hey, everybody. How's it going so far? Good?
Welcome to Stat in NYC. I'm Matt Herper.
And I'm Nick St. Fleur.
I'm the editorial director of events at Stat.
I'm the associate editorial director of events at Stat.
And right now we're trying to get all of you to sit down.
So take a
Not
seat.
to be subtle.
Take a lowdown off. Have fun.
Everybody come on in. We're about to get started. That's the entire point of us being up here.
Yes.
Hello, it's great to see you. We're also going to have these mics out in the crowd.
Yes. So you'll see me roaming around. If you have questions during the sessions, raise your hand. I'll come over. I'll give you the mic. You can ask your question. Be a star for the moment. It'll be a lot of fun, all right?
If you do want a mask in here, that's absolutely encouraged. So it is up to you at this point. And now it looks like the room's filling up. So I think it is time here to welcome you all this evening. We'd like to introduce Rick Burke, Stat's co-founder and executive editor.
Thank you, and welcome everyone. We'd like to make our event special at Stat. So last week we had a dinner in DC for some of our most loyal readers in DC, introducing our DC reporters. And who was next door? We ate at we got a private room at a waterfront hotel near Georgetown. Who was at the private room next to Stat? I'll give you one guess. Anyone know? Right, the President of the United States, and Macron was with him. They came in, and we kept saying, where are all these Secret Service, like, what's going on in here? And right next to us, and we tried to kind of peer in through the thing, but they wouldn't let us. But I told everyone there's real cachet at Stat events. We can get presidents in and all this stuff. Unfortunately, there's no one like that in this audience, as far as I know. I didn't see any Secret Service outside. But what I can guarantee you is this is one of our, I can say it's not our biggest event of the year, but it's my favorite event of the year. I think this is the fifth New York pre-JP Morgan Biotech event that we've had. And it's always just a lively event and with great turnout. Look at this crowd tonight. And part of the reason why I love this event is because it's an opportunity to show off our biotech reporters who, in a very biased way, I can say are the best anywhere. And so I like to see them interact. I like all our reporters on stage. And we also have a great lineup of non-Stat speakers, particularly the focus on Alzheimer's, which is something that's obviously important in the news and something that we've covered very exhaustively in a good way since Stat started 7 years ago. And so I'm really happy that we have that on the agenda today. And I also just want to tell you a little bit about, while I'm here and have the floor, a little bit about Stat's growth. We've just added a bunch of new reporters covering cardiovascular, addiction, commercial determinants of health. We're just adding coverage of disease areas that no other publications are doing. So we're sort of branching out beyond our already intense, aggressive coverage of biotech, life sciences, policy, politics. And we're also, early next year, adding our first international bureau, sending a reporter to London, because there's so many stories that are biotech related and health related throughout Europe. And we see this as only the beginning of Stat's international coverage. So we are growing ambitiously, and our goal always is to provide the best coverage we can. In New York alone, we now have about 15 full-time staffers. That's including our business staff, business team here as well. And I also just want to say, while I'm here, I want to make sure that you all are reading all our free newsletters, including our flagship morning rounds and our the readout, which Damien Gardet, who will be on stage in a little bit, is anchors, and also that you tune into our weekly biotech podcast, the Readout Loud. So there's the readout and then read out loud, if you get that. Anyway, so without further ado, let me turn it over. Am I going to Raul or going to? You can go to me. Okay, I will go to Nick St. Fleur, our associate director, editorial director of events, and let me also just say that Nick is the host of our award-winning Color Code podcast, which is in it's entering its second season, yep. And it's the only journalistic podcast that addresses race and health. And be sure to tune in. Thank you.
Thank you. Thank you, Rick. Now, on behalf of our presenting sponsor, Brunswick, please welcome Raul Damas, head of U.S. Healthcare Practice at Brunswick.
Can you hear me? It's great to see you all. As Rick said, this is the 5th year that we've been hosting this event, which started off, I think, as a conversation at an airport gate in San Francisco when we realized that everybody on the plane was having a great time talking to one another, but that we all were heading back to New York where we normally would spend time if we just had something like this to chat about. And so that year and for the 5 years following, or this is the 5th, we've been gathering here, and it's been great to partner with STAT on this. STAT does a spectacular job, as you all know, of telling the story of some of the most exciting innovations in healthcare. And we at Brunswick work with companies and clients and organizations, including many of you who are here today. It's great to see you, to help organizations tell those exciting stories to organizations like STAT. So I'll keep things brief because you have an amazing panel, including one of our dear friends at Brunswick, Dr. and Dean Lloyd Minor from Stanford Medicine, who is very kind to be here on the East Coast for a change, even though we'll all be going to visit you soon on the West Coast. But thanks again to all of you for coming. It's great to see your faces. I'm seeing more maskless faces than we saw last year, which is nice. And look forward to all the panels here. Thanks.
Thank you. Okay. Well, please welcome to the stage Dr. Jason Karlawish, Professor of Medicine, Medical Ethics and Health Policy and Neurology, and co-director of Penn Memory Center at the University of Pennsylvania. And to moderate, Damien Gardet, our national biotech reporter here at STAT.
That was good. That was good and theatrical. So, Jason, we have you here. Obviously, from your background, people can assume we are 1 week removed from the CTAD conference, where the mood was quite different from previous Alzheimer's conferences. In fact, the last time we spoke, I was there in the sort of sticky with champagne hallways of the Hilton, and you described what we had seen Tuesday night, the ASI presentation, as a shot heard around the world in Alzheimer's disease. What did you mean by that?
The revolution, the
you can use mine.
There we go. No? No? Come on.
The biomarker.
Maybe it's you. Is there something?
Maybe it. The bingo. All right, that's like Alzheimer's. Couple tie, couple shots on goal till finally we hit it. The biomarker revolution has begun. The biomarker transformation of Alzheimer's disease began on Tuesday, the 29th of November in San Francisco. It was a historic event. It is a historic event.
So what does that mean in practical terms? Obviously, the data. I think the sort of cutesy phrase we've all settled on. It was the first clearly positive phase 3 study of an anti-amyloid agent. What did you see that, to you, sets off this revolution with respect to not only understanding the disease, but the way it will be treated going forward?
Yeah, so the study was compelling. You saw consistent results across primary and secondary endpoints, even on measures like caregiver burden and patient self-rating of quality of life. And as a clinician, that was a lineup of results that was spectacular. But what really was provocative, what really is provocative, were the changes on the biomarkers of the disease, changes on beta amyloid and changes on measures of tau. And so what you're beginning to see now, well, this is, I think, an example of an unusual kind of clinical trial. So clinical trials in general are designed to treat disease, to show that an intervention treats or doesn't treat. Sometimes a clinical trial does another thing, which is transform the way we define a disease. And I think that this trial was, and others that have unfortunately failed, were and are efforts at redefining Alzheimer's on the basis of a biomarker test or tests. It's the beginning of that. Obviously, the final goal would be to completely separate the clinical expression of the disease from the biomarker expression and allow a biomarker-based diagnosis to label a patient and lead to treatment. So this was the beginning of that. And like I said, it's a revolution. Remember, what revolutions do is they sometimes shed blood, and this one's going to shed blood, both literal and figurative. We should probably talk about that.
Yeah, what's the literal blood? That'll be
Yeah, okay, I knew you'd go for that, like a good reporter. Go right for the, don't bury the lead. The literal blood will be blood tests. I think some of the data that was particularly exciting was that blood measures of the biomarkers of the disease, amyloid and tau, followed in the direction, favored treatment. I mean, it was greater than chance compared to placebo. And of course, the attraction of a blood test is just that compared to it is far more efficient than the biomarker tests we have, which are imaging tests that require FDG imaging, so there's a whole lot of built-in costs there. Here it's just a tube of blood sent off to the lab. And my colleagues and I are all of a uniform view that these blood tests are going to revolutionize the way to diagnose the disease and potentially, we would hope, follow treatment. And I think there's a lot of enthusiasm around tau testing in particular, tau being one of the measures of neurodegeneration. A lot of enthusiasm there, yeah. So that's the literal blood, yeah.
figuratively.
Yeah, yeah. The figurative blood's a little more complicated and could lead to some literal blood, quite frankly. Because, you know, you have cancer, you go see a, good. You have a heart attack, you go to a, right. You have Alzheimer's, you go to a, don't be so quick about that. I see lots of their failed workups, as well as my colleagues in geriatrics, as well as in psychiatry. There really isn't a clear path to say, I am an Alzheimer's doctor, beyond a self-declared, I'm an Alzheimer's doctor. And that's going to be a challenge for this drug and other drugs for a long time. America had 20 years to create a workforce to diagnose and treat this disease. So the challenge is how to roll these therapies out into the American system, healthcare systems, plural, in a way that adheres to ideas of rational prescribing. And I think there's a legitimate concern that we have not built the kind of healthcare workforce to diagnose and treat this disease with a drug like this. Drug has benefits, but it also has risks. It probably will engender the need for genetic testing as well to help understand that risk-benefit balance. I think it will, for ApoE testing. So that's a complicated drug being dropped into a healthcare system that just, quite frankly, isn't ready for it. So that's where I think there's going to be some battles. And then, of course, there's issues around cost. The events surrounding aducanumab gave us some hint about the challenges related to cost. There, even value was hard to discern. Here we have value, but still, there's no question there'll be some controversies around cost. And then I mentioned already genetic testing. I'm sort of piling on here. You have a drug where the risks are associated with a certain genetic test. Also, that looks like the benefit as well. So now we're introducing into the diagnostic practices the need for potentially for genetic testing, which of course genes are carried in heredity. So this re-engenders the family into the decision making. So lots of interesting issues that I think are going to keep us quite busy. But I'd rather face these issues than the way things were on the 28th, which, well, we knew about the press release. But, you know, I'd rather have these issues than the alternative.
Well, kind of that point, and I did want to mention, if anybody has questions, you can raise your hand and someone will find you with a microphone. But in the meantime, people may know you not only for your work, but also for your writing. And I recall, at least, it was in May 20-something of 2021, you wrote an opinion piece for STAT saying that if the FDA approves aducanumab, as it was then called, I won't prescribe it.
Right, and they clearly didn't listen to me.
Well,
but I think
you weren't alone, apparently.
I know.
Yeah. So we now stand, that was about 1 week from the FDA approval date from that drug. We now stand
Yeah.
about 1 month from the expected FDA approval date for lecanemab. I gather from what you're saying that you do perceive this to be a useful medicine for
Oh yeah.
some
Oh yeah.
who
Yeah. Yeah.
would
Yeah.
prescribe
No.
it.
No.
How would you approach, what does that patient look like? And how would you approach explaining to them what they might expect from
Yeah.
treatment with this medicine based on everything you know to this point?
Yeah, so this gets back. We're now developing the point I raised earlier about the challenges. What I'm looking for is an individual who has cognitive impairment that's either causing inefficiencies in their daily life or causing some degree of disability. The former describes what's called mild cognitive impairment, or MCI. The latter describes mild stage dementia. I can do that. I'm pretty good at those diagnoses. Those are challenging diagnoses to make. It's easy to diagnose advanced stage dementia, but MCI, mild stage dementia, those are tougher diagnoses, number one. Number two, I'm going to be keen to looking very carefully at imaging, particularly for signs of microhemorrhages already in the individual, which are very common. The more we look, the more we find in individuals with Alzheimer's. Strike that, they're common. And then that conversation is going to be around the role of understanding one's genetic ApoE status, given the risk-benefit issues that surround the ApoE status. So those are some of the entry conversations. How long does that workup take for me, typically? A new patient workup, done well, gathering history from both the patient and the family in order to arrive at that. That's about an hour of my time. Yeah, about an hour of my time. But I'm enthusiastic about the opportunity to prescribe it. I'm concerned with its wider use in practice.
Was there a question I forgot? Right there. Do you have a microphone? I can't. That's okay.
I got.
The previous Alzheimer's or dementia medication was lecanemab and aducanumab, MAB. They showed that, as you stated, side effects, brain swelling and small hemorrhaging in the brain. And the rating was 0.45 points on an 18-point scale by the CDR-SB scale. What is the new drug? What is that on the scale?
Yeah, so the primary endpoint for lecanemab, as has been the primary endpoint for now, for the last several years for clinical trials in the Alzheimer's space, is the clinical dementia rating, which has both an ordinal categorization of 0.5123, normal, very mild, mild, moderate, severe, and then you can add up the ways you arrive at that with this a measure that goes from 0 to 18 called the sum of boxes. It is not used in clinical practice. If you asked, I don't use it in clinical practice. We measure it in research. I think that's it's an example of one of the challenges in the Alzheimer's space. The languages of benefit of this disease are very opaque. You know, heart disease, death, blood pressure, glucose, they're all very transparent. The CDR will not be used in clinical practice. So 0 to 18 scale, the population in this study had a range, don't quote me, between like say 0 and 6, mean of 3, because they were very mild, so they were sort of piled up at the end. So I always find the conversation, 18-point scale, 0.45 change. Well, but everyone was clustered between roughly 0 and 6 with a mean of 3. Anyway, so that was the main endpoint, primary endpoint, powered to that. And then the secondary endpoints were measures of cognition, function, as I say, caregiver burden, patient quality of life. And what I think was very encouraging was how all the measures of benefit lined up. Often in these studies, what we've seen are these sort of noisy, nothing on the primary but a little on the secondary. Here we had an unusually clean lineup of the benefits. The point, and I'll wrap with your question, we haven't developed the point around the risks of the drug, which is these microscopic hemorrhages in the brain, which can sometimes also can lead to edema, swelling, and if not detected, can certainly cause disability and be rather symptomatic. So there's a need now for frequent MRI imaging early in the course of dosing the drug. Again, introducing around issues around the challenges for clinicians to use this drug. Very novel drug in our practice. Going to certainly require clinicians to get up to speed in how to understand and talk about this risk-benefit balance, how to use the drug. I'm on a bit of a ramble, but if I was at FDA, I would be convening an advisory board to ask questions to help me guide whether I need a risk evaluation and mitigation strategy for this drug. Because you want to roll it out in a way that's going to be prescribed rationally, and you're going to get data about its safety with confidence that those people who are prescribing it know what they're doing. And a REMS can get you there, I think, and it's been used with other drugs in ways that allow them to get out there and be used and not pulled and recalled because of sloppy use and whatnot. So that could be a way to get this out there, get it used, gather some more data, and begin to transform clinical practice.
Damien, we have a question over here.
Okay.
Hi, my name is Tom. You mentioned amyloid imaging versus this blood test.
Yeah.
Amyloid imaging is very accurate, but it's very expensive. Versus these blood tests, you said you gave a lower standard that was more like significant versus a placebo or something like that. What do you see as the role of the amyloid testing in blood down the road? Do you think it'll be something that'll be widespread, something everybody takes?
Yeah, you know, it's
confirmed.
like you know, you turn 50, you have a kind of a miserable party and you get your colonoscopy. Will we arrive at a time where testing for Alzheimer's biomarkers based on age or other? That's a plausible future, but let's focus on the now and the next several months. I would not, and this drug needs to be prescribed only if you've shown you have elevated amyloid. And one could imagine a set of, an approach to this that sets a very sensitive blood test to say, oh, you might have elevated amyloid, and then confirms it with imaging. And in that way, substantially reduces, the burden upon your PET scanners and their cyclotron and whatnot. And I think that's a very plausible future. I think a lot of folks are wondering what would be the role early on for measuring tau as well, and then following tau, because it's a measure of neurodegeneration. I think there's a lot of very keen interest in that. But we love our tests, especially in America. Physicians are a little like teenagers with a keg of beer. They only stop when the keg runs out. Tests are like that. Excuse that piled-on metaphor. But we should be very keen to look at testing also that helps automate and gather data about the brain's function, because that's in the end what matters. So testing that will gather functional and cognitive information about a patient that is separated from the pencil paper testing and whatnot in the clinic, which frankly is just a noisy, messy operation once it's out in wide clinical practice. So imagine a future where, courtesy of this device, where's my cell phone? Oh no, it's right here, it's okay. That can begin to gather data about how my brain is working. And there's a host of technologies that effectively monitor our brain, starting, for example, with the car, which monitors how far we're driving and whatnot. So we might envision a future where thoughtful technologies allow us to detect and monitor cognition. And there's lots of that work already been going on. Yeah.
Yeah.
Damien, we have a couple more questions. We have one with this lady in the front, and then we have a gentleman in the back.
Wonderful.
Thank you, Doctor. You mentioned there's different risk-benefit associated with ApoE status. Do you mind elaborating on that? What are your reactions to the different results that we've seen by subgroup, and how would that impact your treatment decision?
Yeah, no, I mean, we've known that if you're an ApoE4 carrier, everyone has either 0 copies, 1 or 2 copies of the E4 gene. We've known since the long time that each 1 to 2 copies of the gene raise your lifetime risk of getting Alzheimer's disease, although certainly ApoE4 non-carriers develop the disease. There were a proportion of them in the study. And with these drugs, we've found that ApoE4 carriers are more likely to have those microhemorrhages and edema. And this study showed that. That was a finding that was not a surprise. The forest plots, and I know you want to look at the forest and not the trees, but there were a couple trees that were always the same kind of height in that forest, namely the non-carriers' rate of benefit compared to the 1 copy, 2 copy had a sort of dose response, dare I say, decrease, albeit still over the identity line in benefit. And so what you have is the hint of maybe the E4 carriers, as they are not obtaining as much benefit, and you also have the signal that E4 carriers have a greater chance of risk. Now, I think that's a decision for the patient to make, but I think they need to know that you have a gene that raises your risk, and also may limit, dampen, I'll need to work on my description there, benefiting, and that's how I would think about it. But that's a case-to-case basis, and again, and that's where I think, you could see a value for a REMS program around this drug.
Damien, a question in the back here.
Yeah, thank you to the panel. Just a quick question, assuming aducanumab ran into a problem with CMS, do you think that the results are clinically meaningful, that CMS will have no issues reimbursing it? And if so, what should be the level of reimbursement, generally speaking? Thanks.
$5. No. I think the Biogen story showed us where drug prices come from. I think when they cut that price from 60 to 20, that was a very big reveal. We'll stop at that point. Is the drug reasonable and necessary for clinical practice? Again, if the data that I've seen describe a drug that's reasonable and necessary for clinical practice, okay, but I expect FDA to take a deep dive and take a close look. But if those data obtain, but then I think the question that CMS and FDA ought to work on together, dare I say, they're in the same department for God's sakes, would be how to roll this drug out in a way that's rational. And this gets back, right now CMS is on record of wanting a study. They're open to registries if the drug has had a phase 3 approval. Registries are great, but CMS registries are a bit clunky. Maybe they'll make one that isn't so. I would like to hear CMS and Medicare CMS and FDA having a conversation about a REMS, or how can we design this registry in a way that gets the drug out there, but also gets it prescribed rationally and safely. That would be a really great moment in American history. I think the one thing we learned from the FDA decision on aducanumab was the lack of any communication between these two agencies, or departments within the agency, which really was kind of disappointing on a number of levels. So, and I'll stop there.
Well, that is exactly the amount of time that we had. So we're getting off on schedule, which is rare for these
Brilliant.
events.
It was a lot of fun. Thanks a lot.
Yeah, thank you all. Thank you, Jason.
Next, for a sponsored discussion, please welcome to the stage Dr. Lloyd Minor, Dean of Stanford University School of Medicine, and Matthew Herper, Senior Writer, Medicine, and Editorial Director of Events at STAT.
Hey,
How are
Lloyd,
you?
welcome. How are you?
I'm doing great.
You're the one who had to fly here. I just had to take the subway.
It's good to be here. It's good to be here with everyone here and with you.
It's great to have you here. So your business, just about every business it feels like we talk about here, your business is going through some pretty profound change. We've seen in COVID, like, what has the collaboration that you've seen there meant? What's different now as opposed to 4 years ago at Stanford?
A lot. I think one thing, there were so many of our faculty who pivoted early in the pandemic, who had not done virology or vaccine-related research, but who had ideas and pivoted to studying either the immune response to the SARS-CoV-2 virus, or novel approaches to developing a vaccine, or novel ways to monitor the spread of COVID. So a lot of innovation that got channeled towards COVID, and I think will remain directed at infectious diseases and better understanding the immune response and the spread of infectious diseases. The other thing that's gone on is that we're seeing a change in the funding landscape, I think, in biomedicine. This began before COVID, the Chan Zuckerberg BioHub, Chan Zuckerberg Science.
Billionaire science, as I would have said.
Billionaire science, right. And of course, probably the first of them all at scale was the Broad Institute. And that model continues to expand and grow. And I think for us in academia and at research universities, academic medical centers, we could say, well, what we did before worked fine. I'm not sure it always did, but we could say that and sort of hope for the era in which philanthropists simply gave money to institutions rather than starting their own institutions. But I don't think we're going back to that world. I think we're going to continue to see the proliferation of both in the nonprofit sector, the for-profit sector, of research institutions with great scientists doing great science. And then for us in academia, it's how do we partner with them when we can, and what's the appropriate relationship between academia.
There's an element of competition too, right? I mean, these, a group or company like Altos, or even Chan Zuckerberg, they're poaching they may not all be able to poach your people, but they're poaching you and your colleagues' people, right? Like, there's a competition for jobs.
Yes, and for talent. Now, Chan Zuckerberg and BioHub, the faculty remain in situ. They remain at their home institutions. And that's been wonderful because it's gotten collaborations among the 3 Bay Area institutions, UCSF, Stanford, Berkeley. The collaborations funded by and supported by BioHub have dramatically expanded the level of interaction among those 3 institutions. That's a good thing. In the case of Altos, it's a for-profit company. And yes, faculty that go to Altos, they can have a 2-year leave of absence. We haven't had anyone yet on the Stanford faculty go to Altos.
Is that a point of pride there?
Well, I hope it says that we enable our faculty to do their very best work. And when I have a conversation with a faculty member about, they're looking at an opportunity, and as a dean, there's only one thing worse than having your faculty poached or being poached, and that is not having your faculty poached, because that means that your faculty really aren't probably worth that much in an open, free market. But when I have conversations with faculty who are considering another opportunity, it always comes down to, where will you do your best work? Where will you be the happiest? Most of the time it's by staying and by making sure that we've made the opportunities available for them at Stanford. Sometimes it's going to be by making a move, and then we should be doing everything we can to ensure they have a successful move, and hopefully we can continue to collaborate, and they will continue to collaborate with people at Stanford.
So what is the appropriate kind of collaboration with these new models, and how does this change? Because you've always had collaborations with for-profits like a Merck or a Pfizer, and there have always been, the first billionaire-funded science effort I can think of is the Howard Hughes Medical Institute, or maybe the Rockefeller, right?
Right.
And that's been as long as any of us can remember. So how does that collaborative relationship change now?
Right. Well, each one differs a bit. So what we've tried to do is to articulate some principles that we would follow as we consider each one of the opportunities or the either nonprofits or for-profits that's arising. And those principles, a few of them are, number one is that our faculty have to have absolute academic freedom. So when we worked with Apple doing a large study of watch to see if watch could detect AFib, the most common arrhythmia, we had to have complete control over that study. And that meant that we were going to publish what we found, period. So that's one principle. Second is we try to avoid exclusive relationships. We want to be open to partnering with multiple institutions. Occasionally, if it's a contract with industry, there does have to be some exclusivity, but we try to do that carefully. Third is that we never want to be just implementing something. We want to feel like the partnership or collaboration really leverages in a beneficial way for us and for our partner the unique strengths and abilities of Stanford. If it's just another trial site or something like that, then we have to question, is that really an appropriate use of our faculty's time and resources? So those are some of the principles: academic freedom, avoiding exclusivity. Does it fit into the sweet spot of what we feel like we could do in a transformative way? And finally, and probably most importantly, does it enable our faculty to continue to succeed? And associated with that is, does it protect our students? And we don't want students to get caught in
Expand on that a little bit. How does this endanger and/or protect students? What's that role?
Well, in industry interactions, you have to be careful, because if there's industry-sponsored research being done at Stanford, then if a graduate student is going to be involved in doing that research, then we've got to make sure that that graduate student has full control over the project, because they have
the right to publish.
And the right to publish, exactly. For postdocs, it's a little bit different, but everything has to be disclosed and understood by the postdoc up front, including what their role will be on any IP or relationship coming out of it. So I think their transparency is the best principle, and making sure that people who go, that trainees who go into these relationships are doing so understanding what they are, and that they have the freedom not to do it, that they're not being coerced into doing it or feeling like that their position is in jeopardy if they don't, sign up or join this industry collaboration or other.
Faculty have gone to industry for a long time.
Yep.
But it seems to me that the membrane's gotten more permeable. How is a career, if you're talking to one of those grad students or postdocs now, how is their career now different? And what do you want them to know about what this new collaborative environment means for them? Are they going to move back and forth? Is that conceivable? Used to be if you went to industry, you weren't coming back.
Yeah. And I still
You've
think
had some great work from Peter Kim, who is famously, some people might not even remember, but was the head of R&D at Merck in the COVID pandemic. So how does this look now for?
Yeah, of course, with Peter, though, he was at MIT and the Whitehead Institute and built a very distinguished career as an academic before he went to Merck. And then
right, but I'm saying it used to be you would go to Merck and then
you wouldn't come. I still think, our advice is if you think you want to have an academic career, that's great, number one. Number two is it is best, not say it couldn't be done in reverse, but it is best that you actually launch that career after your postdoc rather than going to industry and then trying to launch that career. I'm not saying it's impossible to do it if you go to industry, and sometimes there's a reason related to a project, but the standard way and the proven way for success for someone who is thinking, well, maybe I'll do industry at some point in my career, the proven way is to establish yourself in academia first, get tenure, and then, we allow our faculty to take up to 2 years of a leave of absence to work in industry, during which time, their job is protected. After 2 years, they either need to come back or they have to resign their faculty appointment. We can rehire them later on, but we can't lengthen that leave beyond 2 years.
So what does this mean for innovation? What does this new world of, what can people expect that's different from before in terms of what emerges?
Well, I think there are a few things, and although there are differences among these various entities, whether or not it's the Broad or it's the new ARC Institute that Patrick Collison funded, or
Stripe, right? Like credit cards funding science. It's great.
Exactly, which is a nonprofit. The ARC Institute is a medical research organization nonprofit, or the for-profits like Altos. In every case, the principle is one that's understandable, and that is so much of biomedical advance today is dependent upon bringing people and approaches together in a coordinated, synergistic way to tackle really gnarly problems. And in an institute type of environment where you're doing targeted recruitment, you need someone who's an expert in imaging, cryo-electron microscopy. You need another person who's a protein chemist. You can put together those teams sometimes in a more facile way than you can in an institution like Stanford, where, for example, when we're doing searches for faculty, we tend to always do best athlete searches. We do broad area searches. We try not to be really specific in a search. It would be rare that we would say, we want to find a protein chemist for a tenure track position with an interest in, G protein coupled receptors. We may announce that we're interested in someone who studies the structure of receptors and their function. And if it happens to be that the best person is focused on GPCRs, then that's fine. But we really do want to recruit the leaders in various different fields. Organically, then they group themselves together collaboratively. Most of these institutes are actually, in a thoughtful way at the time they're doing recruiting, hiring the people they need to form teams. Now then, are they going to work well together, and will they accomplish, in an organized way, what has always occurred in a more organic way? That remains to be seen.
All right. Well, thank you very much, Dr. Miner. It's great to have you.
Thank you.
And unfortunately, our time is up.
Okay. Thanks a lot, Matt.
Thank
Good
you.
to see you.
Bye.
For our next session, please welcome to the stage Ivan Chung, Chairman and CEO of ASI Inc., Global Alzheimer's Disease Officer, and Senior Corporate Officer, ASI Company Limited. And to moderate the discussion, Jason Mast, a General Assignment Reporter here at STAT.
It's great to have you, Ivan. Thank you very much for coming. Are these working? Yes.
Yes.
Okay.
Things working, yeah.
All right,
yeah.
there we go.
Thank you, Jason.
Yes, we just had a great conversation from a few minutes ago about some of the challenges that are going to come from putting out this therapy into a world that is maybe not used to having Alzheimer's therapy, and certainly not used to having an Alzheimer's therapy that requires one to have all these kinds of scans and infusions. Tell me a bit about how ASI is thinking about that and how they're trying to deal with that challenge.
Is a great question. I think Dr. Karlowicz mentioned a lot of important points earlier. So it may also be helpful to trace back to history of Alzheimer's disease a little bit, because this is actually not the first go around for ASI to think about such issues in the real world. Back in 1997, when we launched Aricept or donepezil, that opened up the possibility that these patients could actually be helped with pharmacotherapy. Same question back then, how do we implement that in the real world? Of course, that's a symptomatic treatment, and you'll be surprised, some of the biggest breakthrough at that time was things like developing the clock drawing test, developing the MMSC, things beyond the clinical trial success. So likewise, this time is going to be very similar, of course, at a level a lot more sophisticated. So it's not lost on ASI that the real battle to fight for patients and families actually start now. So we take a very, I would say, localized approach. We look at every country, and within each country, we look at each community and try our very best to map out the patient flow and the patient journey to figure out step by step, as you heard earlier, from screening. What can we do in screening in this local community? What can we do in amyloid confirmation in this community? What can we do with regard to infusion in this community? And of course, all the monitoring, MRI monitoring and other kinds of monitoring we have to do in each community, and find ways to work with the stakeholders in each community, whether we're talking about the health systems, the clinics, the infusion centers, the efficacy partners, who are very, very important, the efficacy partners, because there will be many questions from families. They may go to their doctors, they may call the local efficacy organizations, and of course, they may call us at ASI, and we have to address all those issues. So it's tremendous amount of work I can't even describe in words in 5 minutes.
Do you have a sense of like how long, like you might get approval in the next month or two, do you have a sense of how long it would take to go to a point where all that is ready for, say, a majority of Alzheimer's patients who are eligible to get that therapy?
That's a great question. So we can debate about the CMS policy, but the CMS situation, in a way, kind of forced this, launch to be a staged process. So at accelerated approval, of course, the CMS is very clear. Essentially, no access for the Medicare beneficiaries until we have the full approval. And as we said many times, within days of the accelerated approval, we will hit the button and send in the dossier with the Clarity AD data to the FDA for full approval. So there are going to be many months between the accelerated approval and at some point CMS making the decision, hopefully, to allow more open access, and there will be many months. Those many months will, fortunately or unfortunately, allow ASI and all the local partners to really map out and figure out and be ready for that patient journey. With that step in mind, I personally, I mean, with all my colleagues in ASI we're looking at potentially probably 3-4 years out, to be able to get there. And the blood test was talked much about earlier, because even in 2 years, let's say in 2024, for example, that a lot of these health systems kind of figure out the way to do this. Still, PET imaging and CSF still is not conducive to really broader, driving the patients to treatments like lecanemab. So we do believe that having wide adoption of blood tests will be that kind of a final draw to get people to these therapies like lecanemab.
And when you say 3 to 4 years, you mean for the sort of ultimate rollout, not for the CMS?
No, I don't mean, no. I will not be doing my job. If it takes 3 to 4 years with a CMS, I should not be here anymore. I don't mean that at all. And about CMS, this is interesting because Dr. Karlovich was talking about the FDA and the CMS working in the same department, and I think progress is being made there, from my observation. And at ASI, we're encouraged with what the FDA is doing. We're encouraged also by what the CMS is doing right now. You see, I think I can't say much more than that, but I think you are seeing, the Department of Health definitely taking notice on what's the right thing to do for this class, for these patients and families.
And part of what both agencies will be looking at is sort of the safety questions that we also heard about, and how you're going to manage that risk in the real world, especially for all patients, but especially for these homozygous patients. Can you talk a bit about how you guys view that and what you think it's going to look like after launch?
Yeah, I think first and foremost is fully transparent with all the data. At ASI, we always have this principle of showing more than showing less. We don't like to control narrative. We like to show as much as possible, and then have a healthy debate with the stakeholders in the medical community. And with regard to safety monitoring, we are fully committed to strong educational resources to support families and physicians to have the appropriate conversations. And if you look at this trial, this is actually not a broad population to start with. As you heard earlier, this is the MCI due to AD and early AD dementia with amyloid confirmation. This is already a biomarker defined, specific, not a big population. We're talking about very specific population. And in the trial, you see that at least from our view, and we welcome others to have their views, we believe the benefit risk profile does exist across a broad range of individuals in that trial. And of course, it's going to be up to the families and their physicians to make a decision. If they decide not to get on lecanemab, we respect that. And if they decide to get on lecanemab and they happen to be 2 copies of E4, I would really, and I think all of us in ASI will really hope that the physicians and the families can commit to monitoring. If you don't commit, maybe not the right decision, not the right therapy to go on. But I think overall, you look at the Clarity AD trial, the positive clinical efficacy results, the positive underlying biomarker measures across all types of pathophysiology, the positive impact on delay of disease progression for individuals who potentially stay longer in a milder disease of Alzheimer's, and last but not least, the improved quality of life and caregiver burden. You have all, the convergence of evidence here. We don't. This is not a cure. By no means is a cure, but just like Aricept 25 years ago, that sparked, the idea that you could help these individuals with pharmacotherapy. This one, I believe, sparked, and we at ACE, believe, it sparked, this, transformation to a biomarker-driven world of Alzheimer's disease, and something has to be there to spark something. I think this is what it is.
And Matt and Nick are going around. If anyone has any questions, just raise your hand. Would love to hear from them. I think we have a couple
right
here up front.
Right here in the front. Say your
Thank
name,
you.
go right ahead.
Rich McCary, thank you. I wanted to ask, first of all, with the potential for accelerated approval, then followed by full approval, we saw the problem that Aduhelm had with just this broad label that came out at first. Do you see yourself and the company influencing what the label is on the accelerated versus the full approval and what that might have in implications to those prescribing it or to reimbursement? And the second part of the question is, given, I was going to say, neurologists have atrophied in treating, but really the muscle's never really been exercised, how big of a barrier do you see the number of neurologists in the system to actually diagnose and treat as sort of a bottleneck to getting this to patients? Thank you.
Loaded questions. We've said that many times at ASI. Our view is accelerated approval is never our goal. Our goal has always been the full approval. That's what we've been always looking at. It just turned out that the accelerated approval will allow us to get to full approval faster. It's really a regulatory strategy approach, and which is being copied by other companies. And so the label we're looking at is really the full approval label. It's an interesting topic because, we, of course, believe in the precision medicine era. We believe in the biomarker-driven era. We want the label to be as specific as possible. At the same time, you hear stakeholders, efficacy groups out there saying, hey, ASI, this is a free country. You should not force us. It's up to the families and the physicians. So I think we are balancing that. I think the FDA is balancing that also, and the CMS is balancing that. So it's going to be an interesting development going forward. If we have the vote at ASI, we prefer a very specific label. To the last question, yeah, you're right. And, we had some of those experiences with Aricept launch in the past. So it's very different this time. It's a different type of modality. Just the patient journey is completely different, but we understand your point very well, and it's worth fighting for. So thank you.
We have another question here in the front.
Hi, my name's Jenna Lee. You did mention leveraging local partners, and you do have a partnership on lecanemab with Biogen, who is pretty well entrenched in the CNS space here in the US. So to what extent do you plan to leverage that as you think about the commercial rollout?
Yeah, so, we're open book in ASI. And of course, Biogen is our partners on lecanemab, and both companies have a shared, strong desire to serve this community. So we look forward to work together to solve those challenges. But again, as I mentioned, no experience is more important. I think the word neuroscience, we actually, sometimes we don't want to use that word. It's a broad space. It's a very, very broad space, so we tend to be specific. Sometimes when folks use the term dementia, I often would say, wait, let's be more specific here. We're talking about Alzheimer's here. So, our decades of experiences working with local partners across the country in Alzheimer's will, I think, pay a big dividend this time. Thank you.
Jason, we have another question right over here.
Hi, my name is Catherine. I'm a pre-medical student interested in studying neurodegenerative diseases. So my question is, I remember this summer I read an article about potentially falsified Alzheimer's research that challenges the amyloid hypothesis. And of course, there are many, many studies that do support the relationship between amyloid and Alzheimer's disease. I am wondering about the potential correlation between the reduction of amyloid and the long-term cognitive and functional aspect for the patients. So, how do you envision the, also the relationship between this exciting results of lecanemab's efficacy with the long-term aspect of the patient's actual cognition and function?
Thank you. First of all, the phrase amyloid hypothesis, again, is a very loaded phrase that could mean many things to many people. What we can only say is what the trial is designed to do. The trial is designed to show if you impact the amyloid cascade in a significant way, does it or does it not impact cognition and function, and then of course, do they translate into quality of life and caregiver burden reduction? And I think the answer is definitively yes. From the clinical trial, that's the best I can say. Thank you.
We have another question right over here.
Hi, Marco Kinsella. I'm a statistics professor at Central Connecticut State University in Connecticut. I'd like to know how large was your sample group in testing this drug, and what percentage of the sample group profited, slowed down Alzheimer's, and also did you do a demographic sampling, African American, Hispanics, et cetera, because you know that the drug may only work on a certain sector of the population. Does it work on all ethnic groups?
Great question.
Thank you. In this so-called Clarity AD phase 3 confirmatory trial, the data presented last week has 1,795 subjects. A very large trial, very simple trial, just 2 arms, placebo or active treatment with lecanemab. On the lecanemab treatment, 1 dose, no titration. You get the same dose from the beginning until 18 months, as simple as you can do. And I think that helps the statistics also. And actually last week, we showed the results using additional statistical approaches to confirm the sensitivity and the robustness of the primary statistical model, and they all looked very consistent. To your point about subgroups of individuals, one goal of this phase 3 trial is to break it's true, a lot of the Alzheimer's trials don't have sufficient diversity population, and that's what Clarity AD, we designed this to be different. We designed Clarity AD to mimic the Medicare population as much as possible, and we do that from 2 angles. One is in terms of the diversity population. Not perfect, but we did all right. We had 4.5% African Americans in this trial, not the 9%, 10% in Medicare, but not 0 like many other trials. Hispanics, we actually enrolled 22.5%, so actually overrepresentative compared to the Medicare population. So this is, I think many people look at this trial as kind of a good example to do how to enroll diverse populations. Take a lot of work. All the local outreach initiatives, find the right investigators, the right sites, and reach out to faith-based groups, do a lot of decentralized trials through technologies. So some of the assessments can be done at home, not extra burden to come to the sites. Another dimension of the representation of this clinical trial is to be a bit more relaxed on the inclusion criteria on comorbidities. So, diabetes, hypertension, you can be on anticoagulants, okay? Risky move, but we did it because we want to know what's going to happen if you're on anticoagulants. You can also be on aspirin, many medications you can be on. And still, you look at the benefit-risk profile. We talked about anticoagulants, but even you're on anticoagulants, yes, you have those safety signals that you have seen, but if you look at the clinical efficacy measure, people on anticoagulants on those subgroup analysis, you see pretty clearly favoring lecanemab also. So that's how we did the trial. Thank you.
Hello?
We have a
Yeah, come
question
back
back here.
Yeah,
Dr. Chung,
Identify yourself.
Jason Karlowicz, good to see you.
I was looking for you. I couldn't see it that far
Yeah,
back.
marvelous job. Job well done. What are your thoughts about working with FDA to put together a REMS plan as you roll this drug out?
I don't think I am at the position to disclose regulatory conversation with the FDA. I hear your point. And what I am going to say is our goal is always the full approval label. That's the goal, because in a way CMS is going to do what CMS is going to do. So I think we have some time to work with the FDA and to get feedback from many experts like yourself on the best way to go forward. That's, I think, the best I could say at this moment. But again, we're open book. We listen to all the advice, and we want to do the right thing, because I think one thing you can see in the lecanemab program is it's very difficult to resist the temptation to take shortcuts. We always understand at ASI, yes, urgent needs out there, but never shortcuts. If it takes time to do something right, we do something right. We ran like a 6-year-long phase 2 trial, which no one would even want to bother with. But that's what we did, and that 6-year-long phase 2 was really the success factor to help design the phase 3. That's why you have this kind of phase 3 that no one else has done. It's because of the long journey we took to do the phase 2. So,
Jason.
Thank you, Jason.
Jason. We have another question over here.
Just quickly, did you stratify your analyses by staging and by sex, and did you see any differences in those stratifications?
There are 4 randomization strata in the trial, co-medication or not, MCI or mild AD, carriers or non-carriers in terms of the ApoE4, and then the regions, the geographic regions. So these are the 4 randomization strata, and last week you saw the data, everything in these across all these randomization strata favoring lecanemab on multiple measures. With regard to gender and others, those are not randomization strata. Now, we chose to show everything, even on gender, age, on ethnicity, race, everything. We showed everything last week, even though they're not randomization strata. So the trial is not powered to show those things, but we showed it anyway because so many stakeholders asking us to show everything because they want to know, so we showed it.
In the last couple of minutes we have here, I just want to ask about price as well. You have the privilege of going through not only the FDA, but also CMS, as you've talked about. FDA never considers price. CMS, clearly Biogen thought they did. I'm curious, has this affected how you guys are going to launch next month or the month after, and how you'll price the drug?
Yeah, that's maybe the biggest question among them all. Of course, by law, CMS not supposed to consider it price. Is an evaluation of the necessity of the medication, but nonetheless, it's not lost on ASI how important this decision is. So first and foremost, we will be fully transparent with our rationale. We have already started making publications on the economic modeling of the value of lecanemab based on our phase 2 data, and we're updating that economic model based on Clarity AD, and we will publish that. We'll be fully transparent, and everyone can debate whether they agree with our assumptions or model or not, but it's going to be out there, and we will definitely consider patient affordability and system sustainability as 2 additional factors in addition to the value to the society. So these 3 factors will put everything together, and our goal is to drive wide access as much as possible. Ultimately, that's our number 1 goal. Thank you.
Thank you very much, Ivan. I appreciate it.
All right. Well, this isn't over, everybody. You know, we've got another big event here. Please welcome an all-star cast of stat, pharma, and biotech reporters. Allison DeAngelis, our biotech startups and venture capital reporter, the queen of venture capital. Damien Gardet, the master of the mellifluous phrase. Jason Mast, the guy who was just on stage and one of the fastest writers I've seen in a long time. And last, but certainly not least, the man himself, Ed Silverman, the legendary Pharmalot. And to moderate, Nicholas St. Fleur, General Assignment Reporter and Associate Editorial Director of Events. Everybody make some noise.
Make some noise, everybody. Hey there. So what's particularly great about this session is that it's your chance to be reporters and ask the reporters up here as many questions as you want. All right, so think of some good ones. We're also going to be talking a bit about, 2023, what these reporters have in their sights, what they're looking forward to. But before we get there, I want to start a little bit with 2022. It's still here. It's still kind of cool, And I want to ask the reporters a bit about...
It's not cool.
I think it's cool. I've enjoyed it. A bit about, some of the stories that they've really liked this year, or some of the big stories that they've covered. I actually want to start with Damien here. Damien, you just had a panel with Dr. Karlwish, and he asked you a bit about, things that bleed figuratively and literally. So I'm wondering, did you have a story this year that maybe drew some blood, whether it be figuratively or literally?
Not literally, mercifully. I'll shout. Or no, now it's working. I got it. Not literally, thankfully, or at least to my knowledge. Working on the internet probably prevents paper cuts or any such blood-drawing things. But I was thinking, actually, listening to Ivan just now, I think that the covering Clarity AD in September, not that we did, I think, really a different or demonstrably better job than the hundreds of publications in however many languages that covered it, but was the most interesting story of the year, in part because I and many of us have written many stories about Alzheimer's disease trials that fail, and they often fail in very interesting and instructive ways, but to have one that met its goals, and then to be able to ask kind of the same cast of characters of experts in Alzheimer's disease and people who follow this, a different sort of question, which is rather, why do you think this failed? What can we learn from it? But what does success actually mean, and where do we go from here based upon what that entails, which led up to last week at CTAD and just having richer and different conversations about Alzheimer's disease, which is refreshing to me, just professionally, in addition to the obvious societal implications of a drug that seems to work. Just selfishly, the novelty is fantastic.
I mean, was that a story or something you thought you were going to cover coming in to this year?
Did I think Clarity AD was going to work, is your question? Like I said, having the experience of so many trials that either outright fail or work with an asterisk, I kind of expected the latter at best, and so I had the pleasant surprise of novelty, in addition to the, like I said, the implications of the actual readout.
Great. Allison, let's turn to you. Okay, so what was a big story that really you enjoyed covering this past year?
Oh, God. There's, a couple. It's been a really interesting year, and actually, just as Damien was speaking, I was reflecting on when I started kind of talking to and covering biotech venture capital was about 5 years ago when biotech investing was very much on the upswing, more and more and more money was going in every year. And, you know, my editors would encourage me to ask about, like, is this a bubble? Is this going to pop? What's happening? And everybody was like, no, no, no. And this was the year, I think, where it was kind of a different conversation, where I had so many conversations with investors this year who were saying, yeah, we're like really like we're tightening our strings, you know, our purse strings. We're being more conservative with where we put capital, and that's just a different conversation than they've been having the last 5 years. And actually, a lot of people who are in the investment side, a lot of biotech investors haven't gone through this kind of economic downturn before. So it's really kind of testing them. Plus, we have a lot of new investment firms that have started in the last couple of years. And so this is a really huge test for them to see if they're going to be able to survive. I think that, outside of that, this year, the last year or two has really been a reckoning for what you should be as a leader in biotech, and what kind of standards should be set for your workplace. Obviously, Jason and I wrote about, some of how the very rich biotech research organization at UPenn, run by Jim Wilson, turned out, and how that's been affecting not only his staff, but also the research collaborations that they've been doing.
I'd love to elaborate a little bit on that. What has this past year showed you is, I guess, bad in a leader in biotech? And then what's kind of good in a leader?
I think, it reflects so much of, all the headlines that we were seeing in, 2020 of, leaders that were pushing their employees to, work harder, work faster, just get it done. So much of what we encountered from those employees was there was a base level of, we are being asked to do work that is just way beyond our capacity, because they just keep on bringing in new research projects and collaborations. And then, I'm trying to remember the circumstances, but talking about like a big ego science, and that there's so many scientists in this world who have gained reputations that have made them a little bit infallible. And we have people now that are saying, you can't treat people in that manner. How you are and how you treat your colleagues and your employees matters. And the ways in which you might, put them down matters, and it will not be stood for.
That's an important lesson for all the managers here, all right? Jason, tell us a bit about, your views. Like, do you agree with everything that Allison was just saying there? I know you guys have teamed up on a few stories.
Yeah, and if you look at the, especially with that Wilson story, one of the major takeaways that we had from it was not that this was not by far not the greatest place in the world to work, and people told us horror stories about there, and every holiday and weekend and that they missed, and 3 a.m. that they were there, and being yelled at on occasion. It was the fact that was disrupting their work, and that was like actually not conducive to the vision that he had, which was a great vision of making gene therapies for rare disease patients. And the remarkable thing was that you talk to people who knew him well, and they would tell you about how much he genuinely cared about these patients. But it seemed at times that he was willing to throw his employees under the bus to get there. And the result is that, you can look at some of the companies that Wilson has founded in the last couple of years, and they're not, there's a variety of reasons for this, but they're not doing particularly well at the moment.
Now, Ed, Mr. Farmalot, what about yourself? Any stories this past year that may have literally or figuratively drew some blood?
Can you hear me?
Yes.
I pissed off the Indian government a few weeks ago.
So hoping to talk about that.
I spent about 6 months with some sources who provided me documents, some of which had not been previously circulated, concerning the machinations in India to eventually approve one of the COVID vaccines. This is made by Biotech. And the issue in gleaning, just going through this real quickly, was that the special expert committee that was convened by the regulator did not follow the usual steps for getting from one sequence of the trial to the other, moving too quickly before having results. And the context here is a larger issue plaguing the Indian government and the Indian pharmaceutical industry, which is to what extent is there proper oversight and regulation. That's come up before, often in the case of manufacturing, going back years. You remember the name Ranbaxy Laboratories, there was a scandal. But it also has played out with questions about the oversight of product approvals. And so to fast forward, after a few months of working on this on and off with people, deciphering all the documents I had and writing it carefully, it came out recently. And I must have written, I don't know how many emails to how many people in the Indian government and on their various committees. Would you like to comment? Trying to get someone to respond. I got no response. Day and a half after the story comes out, one of the ministries puts out a nasty note, knocking the story. But the company also put out a statement that pretty much confirmed the story. So that was fun.
Ed, I'm going to stick on you here. Looking forward to 2023, what are you excited for? What do you have your eyes on? And are there any other governments you think you'll piss off?
Well, India is just a diversion for me, actually. What am I looking for? Well, I spend a fair amount of my time when I'm not doing wonky policy stories looking at pricing, and but I try to look at it through different prisms, and I'm curious to see, in no particular order, what happens with gene therapies approved for sickle cell, because they're really the same drugs that are being explored or tested or used to treat other rare related diseases, but you've got very different populations. Can you price a drug for beta thalassemia up here, but can you price it for sickle cell here and still get away with that, given the need in a Medicaid population and what that does to state budgets? So I'm curious to see how that works out. It's something I wrote about several months ago. Insulin is a bugaboo because it's a problem for folks who still can't afford it. Doesn't matter what the IRA says. You've got commercial insurance that isn't part of that scheme, and because some folks pay based on what the list price is, we still got a problem. We've got 3 insulin makers, but only 2 of them have really actively engaged with programs that are around $35. You get a free soda outside if you can guess the company that hasn't done that. I'm curious to see what happens with the pricing for Alzheimer's. That's another interesting concern, I think, for all sorts of budgets. And with obesity, suddenly we have, I've been around long enough, going back to the 90s, the 1990s, not the 1890s, when there were diet pill scandals and all sorts of issues with fen-phen and Redux and side effects, some of which were not disclosed, and court cases and all sorts of bad, nasty, evil, ugly things. And we've got different types of drugs now, but what's going to happen with the price? What's going to happen with the reimbursement? We're going to have so many people knocking on doctor doors for those medications. Will there really be coverage for everybody? Will there be coverage? How's that going to be decided? What's the reimbursement going to look like? What are all the ramifications going forward? So those are just a few things off the top of my head.
Now, before I turn it over to Damien, we have a question over here in the audience. And again, I'd encourage everyone, if you have questions for us, just flag down my colleague Matt.
Raise your hands. This is a question from Ian Spatz at Murat, Phelps, and Phillips.
Manat Health, yeah, hi. I'm tempted to use my question to ask Ed what kind of coffee he's going to have tomorrow morning. But I won't waste the question. You want to answer that, Ed?
Jack Daniels.
Okay. Ed just mentioned the Inflation Reduction Act, which hasn't been mentioned in any of the panels so far today. I'm wondering, does that mean it just is no big deal? Is that why no one's talking about it here?
I think it's such a big deal we could spend another 3 hours on it, actually. No, I think it's a very big deal, and I think there's still so many unknowns, which I know you know about. I'm curious to see what happens with biosimilar launches. I'm curious, very curious to see if all that saber rattling about products that won't get developed actually comes true. I remember going back to the 1990s when I first started covering this industry. I arrived and, oh my gosh, there's 4 or 5 different cholesterol pills and diabetes pills and blood pressure pills. Well, okay, so if companies going forward don't want to develop the 4th or 5th fill in the blank cancer treatment, that does sound like a crime and a shame, but how much better is it than drugs numbers 1, 2, and 3? So if that's an issue, we're not going to see the extra product, if we aren't going to see the first 1 or 2 tested and developed and ultimately hopefully approved, well, maybe those companies should be looking at doing something else in the first place. So instead of crying about, oh, we're not going to be innovative and we're not going to be developing as much, well, let's break it down and let's see what they really do. And maybe they're right in certain instances. It's very easy to generalize, but in my view, I'm not going to believe anybody until I see what happens. And that includes all the complaints about, having innovation tossed aside.
Damien, 2023, what's on the horizon for you?
Well, actually, the one thing Ed mentioned at the end there about the new crop of obesity treatments or weight loss treatments in general, I think everything he mentioned about pricing and access and the normal trappings of like a launch of a apparently blockbuster drug class, but also obesity. There is not universal agreement that obesity should be an FDA recognized indication, that it is in fact a disease. So this is a class of drugs that runs into not just the normal stuff that we're used to with pricing and access, et cetera, but also like cultural issues. Like this is a treatment that has proven to help people lose weight in a short amount of time, but the actual medical benefits of that with respect to prolonging life or preventing heart attacks or anything else, those are data that are still to come. And so it's running up against what seems like a growing and pretty vocal movement of what I think people describe as like fat acceptance research and cultural issues, which are very real. And they're like, this is on the minds of physicians and patients. And then as they move into the mainstream and win approval, they will probably be advertised. And if like the World Cup is any example, there is like this incredible pot of money that pharmaceutical companies have to advertise. I've seen so many advertisements for multiple sclerosis treatments during soccer games, which is a disease that affects, I mean, it's neither here nor there. We're probably going to be like blanketed by marketing for these drugs that are sort of feeding what is an emerging cultural issue while also running into pricing and reimbursement and access issues, and then also whatever's to come with the FDA. So that just seems like a powder keg for the year to come, and years beyond that too.
Allison, any powder kegs in 2023 you've got your eyes on?
Oh gosh, I mean, powder keg, I don't know. We're talking about
firecrackers then.
Little firecrackers. I think that, I mean, this last week we just saw the first microbiome drug approved. We have another one that's like lined up for approval, in April, and there's, a whole slew of others that are kind of right behind it. And they're all going for the same, disease, the same indication. So that's going to create a really interesting commercial atmosphere. I also, I've always been fascinated by like the 2020 fervor of, into SPACs. And we're hitting that point where, around the start of the year and like into the first half of 2023 and like throughout the year, we're going to see several SPACs, many dozens that will hit their window of you had 24 months to invest this money and you didn't do it, and now you have to give that money back to the investors, which I think will be a really interesting mechanism for this environment. It'll be really interesting to see what those investors choose to do with that money that's now in their pockets again. Are they going to like reinvest it, or are they going to say, you know what, we might have a recession coming, so I'm going to keep this here where I know it's safe?
Before I go to Jason for his 2023 predictions, any other questions here in the audience? Any questions for our esteemed panel over here? No, not seen? Oh, right over there.
All right. Get to see me run.
Got to make Matt work for it.
Yeah, totally. Hey guys, Christiana from Real Chemistry. You guys have been doing a lot of reflecting. I'd love to hear from each of you. What's a byline you wish you had this year?
Ooh, the
great
jealousy list. I'm going to start, well, I'll start with Allison again, and then we'll just go down.
Oh my God, there's so many good. I mean, there was such good reporting this year. I really love. I mean, my colleagues, they're just mine. Our colleagues, their coverage of what's been happening with, how the Inflation Reduction Act came about, what's been happening in Congress. I mean, just like such a fascinating look. And then what impact that's having on trade organizations, such a fascinating peek under the curtain. I also really was interested in, the New York Times had, some of the reporters there had done a piece looking back and tying together what role the consulting firm McKinsey played in several different drug companies, opioid product pushes, and their work either kind of creating raw materials for opioids or what have you, which I had suggested we put out the jealousy list every year this time of year, so you're getting a little preview of my suggestion. This was such a fascinating look at these things don't happen in vacuums. They are often, there are people, there are organizations that are contributing kind of from the sidelines, maybe outside of the spotlight, but are a very real presence in making these types of situations happen.
Damien, real quick, what made you jealous this year?
The first thing that came to mind was the Gawker.com headline where they discovered that Elizabeth Holmes' dog had quietly died, which I think was a shoe leather story. I don't remember exactly how they got it, but it was definitely out there for any of us to get, and that was the first thing that came to mind.
I'm going to toss it to Ed. Any dog death stories you're jealous of?
No, I was going to say the FBI raid at Mar-a-Lago, but it's not my beat. Honestly, I probably should schedule a workup with Jason. I can't recall any.
No problem. Jason, what about yourself? What were you jealous of? And then what about 2023?
This will get on both questions. The story I was really jealous of was a story that Allison's old colleague Andrew Dunn did at Business Insider on the SCID, severe combined immunodeficiency gene therapies that Orchard had developed, and with some academics that are clearly curative. We have a bunch of data now showing it years out, but that are not being made and not being sold anywhere. And so you have, because there's no way of incentivizing it. There's the population is too small, the therapy is too expensive to make and administer, so you have these patients who have this severe disease, and we have curative therapies, truly curative therapies, but we can't close that gap. Which brings me to next year, where we have now we had 3 gene therapies approved this year, I think. Someone can correct me on that number, and we're likely to have probably 3, maybe even more next year approved. And we'll get to see whether or not these actually reach patients, as Ed was saying. And not only do they reach patients, but also do they reach patients in a way that incentivizes companies to stay in gene therapy as opposed to another field where they can sell a pill for $300,000 a year. So I'm really curious to see how that's going to play out.
Wonderful, wonderful. Well, with that, we are out of time. I want to give a special thank you to Allison, to Damien, to Ed, and Jason. And if any of you all have any scoops on celebrity dog deaths for 2023, please hit up Damien. Thank you. I wanted to say thank you so much to everyone for coming out today. A thank you to our speakers, Raul Damas, Dr. Jason Karlowicz, Dr. Lloyd Miner, and Ivan Chung. And I wanted to let you all know that right outside this room, in the reception area, you'll find some folks from STAT and our partners at Applied XL, demoing our new clinical trials platform, STAT Trials Pulse. The platform allows you to get real-time updates about the clinical trials most relevant to you, follow curated feeds, and create targeted watch lists, and overall, separate trends from the noise. Be sure to connect with them and check it out. And thanks again just so much for joining us, to all of our speakers, for taking the time to share your stories and expertise this evening, and to our event sponsor, Brunswick, for making it all possible. Now, there'll be more food, more booze, all of that outside. go ahead, stay a while, and thank you so much for joining us, and continue to read stat.